to three possibilities for this overlapping phenotype. The first one is the developmental branching point that the progenitor cell can give to both T and non-T cell lineage, or an alternative entry point for precursor. So as we know, the FLT3 is priming for the next stage of the DN2A and already entered the T cell fate. And here may be the second entry point that the SPI1 cell is also prepared for their next stage for the DN2A cells. And the third possibility is that the transient induced upregulation of the non-T genes even along the T cell pathway. So that means even though these genes are not expressed in the end stage of the T cell lineage, but actually they're the marker of the transition stage of the T cell development and contribute to the transient wave of the gene expression pattern. So to answer this question and to find the authentic possibility of this overlapping phenotype, the group used the second analysis, which is called RNA velocity analysis, to map the developmental flux of the T cell development. This analysis is newly published in Nature in 2018. And the concept that they use is to use the ratio of the unspliced and spliced mRNA to predict the status the cell gonna be. According to their theory, actually when the cell going to change, both the unspliced and spliced mRNA will change accordingly. But the unsplicing RNA will change at first, and then the splicing RNA changed after that. So for example, if the cell going to differentiation, some gene will be induced. And once the gene induced, the unsplicing mRNA transcript of this gene will increase at first, and then the splicing mRNA transcript will increase the thereafter. So the ratio of the unsplicing and the splicing RNA will have a changing pattern of the ratio, and after that reach a new steady state. And reversely, when one cell is going to repressed or degraded, and some gene is also going to be repressed, and then the unsplicing mRNA will decrease at first, and then the splicing RNA will decrease thereafter. So then they will follow this direction to change their ratio and then reach the new steady state. If we have the RNA sequencing data from individual cells, then we can calculate the ratio of the unsplicing and the splicing mRNA in each cells and map their location in this map. And then we can find which cells in a more advanced step and which appear in a less advanced step. And then we can follow this order to find out the substate and the correct order of the developmental flux. Okay, so according to this theory, this group successfully predicted the developmental flux of the T cell development in their spring analysis map. So here they find, according to the marker expression pattern here, they firstly find the FLT3 ETP population have two distinct states. The first state is here that the cell going to change into another ETP state. And the second state is going to contribute to the DN2 state. The arrow just indicates the changing direction. And it's very interesting that this population also have two status. One is going to change, and the other one is maintain their static status. And this population is labeled by the BIRC5. So it's highly possible that this group of the cell that remain static maybe have a high capacity of proliferation and contribute a lot to other progenitor cells. And here is also very interesting to see how these two markers are overlap. Actually, the CD7 positives are the population that mostly contribute to the DN2 cells. So This is confirming the third possibility we have raised before. The CD7 markers is more like a transiently expressed marker of the transition stage of the T cell lineage. The expression of the non-T cell marker maybe also means the multi-lineage priming of the progenitor cells that still have the multipotency to become into different lineage and also have the possibility to change into the T cell lineage here. They use the pseudotime analysis to further map the trajectory the cell follow. Because they failed to use the unsupervised clustering analysis to map the correct order of the trajectory, so here they use the curated genes that used for the supervised clustering before to establish a landmarker here. This is the expression pattern of all curated genes they used, and we can see some of the genes that expressed highly that indicate the cluster of the stem cells, and here are some genes that expressed later in the mature stage, so that indicates the cell of the end stage. And here they use the DDR tree analysis to obtain a connected developmental trajectory and pseudotime staging of the cells based on the curated instructive cell gene list mentioned in sick fish. And by following the pseudotime analysis, they're easily to find out that 11 clusters of the cells follow the pseudotime and express the different markers. Here is all the genes they detected by the single cell sequencing. And again, the column means cells and the row means the genes. And if we look into this gene expression pattern, and then we can find here are several stages that follow an order to change. So firstly, it's very easily to identify the group that is ETP and the DN2. And also they find several intermediate stages here that may review the transition from ETP to the DN2 cells. So according to the expression pattern here, we can find the cell actually they didn't change in the monotonic increase or decrease pattern. What is the monotonic increase pattern? For example, the gene only appear at this way but not appear in most clusters. So that means the cell is monotonic decrease along the development. And this is a gene that only appear at the first stage of the cells, and after that they disappear totally. So this is a monotonic decrease. But except for these two clusters of the gene, we also find several intermediate waves of the genes. For example, this cluster of the genes, they appear at stage 2, cluster 2 cells, and then disappear at cluster E. So here is more like an intermediate wave. They appear after the stem cell gene revoke and disappear before the end stage appear. And this is also answer our previous question. That is, the CD7 marker actually, even though they didn't express that in the end stage of the T cell lineage, so that's why we call them non-T cell lineage, but actually they appear during the transition stage and then contribute to the priming of the T cell lineage. So here we are trying to interpret the intermediate wave by looking into their expression pattern and to see whether they are under the regulation of the TF. That is very important for the T cell lineage. Here is one study that published in 2018, and to find out the regulatory mechanism under BCL11B and the PU1. That is the transcription factor important for the T cell lineage. And by perturbation experiment, that study reports 763 genes that are regulated by these two transcription factors by knockout either these genes or these genes. When they map the 700 genes into their single cell sequencing data, and they find 214 genes are pseudotime indicator genes within these 700 genes. That means there's a lot of the genes are under regulation of these two transcription factors. So they trying to digest these genes into several clusters and to see how their expression pattern are correlated with these two transcription factors. And firstly, they find the PU1 positively regulated genes in the early transient wave, such as the orange cluster, and negatively regulated at late cluster, for example here. Considering that the PU1 are expressed at early stage of the T cell development, actually this gene's expression pattern is highly consistent with its own expression pattern that is active at earlier stage and repressed at late stage. Reversely, they find a lot of genes are repressed by the BCL11B at earlier stage, such as the orange and the green, and are activated at late stage, such as here. The BCL11B are expressed later at the T cell development, so the activation and repression of these genes are also consistent with the expression pattern of the BCL11B. The consistent expression pattern of these genes with their upstream regulator suggested that these genes upregulated in the intermediate wave are expressed within the T cell lineage specification pathway, and their expression are truncated by the landmarkers transcription factors of T cell development. After the group have obtained a lot of analysis data from their sequencing, they need to further confirm their analysis by the wet lab experiment. So finally, they use the in vitro cell culture and the single cell sequencing from the newly isolated cells to confirm their hypothesis. So the hypothesis that is the intermediate expression wave they are very curious about may be a succession of the transient regulatory state, such as here, or computational artifact of forcing branching genes expression changes into a single pathway. To answer these questions, they are trying to isolate ETP cells from the thymus again, but this time they use some new markers to identify this cell in less mature and more mature stage, and trying to see whether these markers they identify from their single cell sequencing data can use to identify the substage. Based on their pseudotime analysis, the cell surface marker HAS, which is also called CD24A, was predicted to be gradually upregulating during late ETP stage. So as we have said before, the FLT3 is the early marker of the immature ETP. So according to their pseudo analysis, the CD24A is more like a marker that appear after FLT3 and indicate the more mature ETP. And the expression of the HAS is followed by the FY60 upregulation. So the pseudotime of these three markers should be FLT at first, and then CD24A, and then LI60. So they use these three markers to isolate the cells and then culture them under the T induction and the non-T induction medium for 4 to 7 days. And then they sorting the cell to see whether they show different speed toward the T cell. And also they trying to use the cell hashing tag and pull the 10x single cell RNA sequencing to sequence this population and to see whether the three markers marking cell population can recapitulate their trajectory predicted by single cell analysis before. Firstly, they use the in vitro cell culture analysis to see whether the cell can review different developmental potential and speeds toward the T cell lineage. Here is 6 population they use the marker to identify. After the induction, they find actually this population of the cell indeed show the lowest speed toward the T cell lineage. Here is the demonstration of how to identify the different stage of the cells. So from the ETP to the DN2A, the expression of the CD25 should be upregulated. And from the DN2A to the DN2B, the expression of the BCL11B should upregulated. So according to the flow cytometry analysis here, we can find the cells can only enter the DN2A stage, but this cell have already entered the DN2B stage. So according to the expression pattern of the markers here, we can find the speed that toward the end stage of the T cell lineage increase. That means the markers identified from the pseudotime analysis actually reflect the trajectory the cell takes along the T cell development. They also replicate the experiment for 3 times, and every time the cell can recapitulate the pseudotime. The ETP population 6 have the highest speed towards the end T cell lineage. According to the theory of the development, when the cell go into more mature and differentiated status, then they will lose the multipotency, which means they are more restrictive to one cell fate but cannot convert into other cell lineages. So they also culture this 6 population of the cells under the culture condition toward non-T cell lineage, such as the granulocyte, macrophages, and the NK cells. After they culture the 6 population and they find only the FLT3 positive cells can give rise to different kind of the cell, but the FLT minus cell fail to give rise to other lineage except for NK cells. So based on this, we know that when the FLT3 is there, and then the cell is more multipotent. And when the FLT3 is turned off, then the cell is more restricted to the T cell lineage fate. The previous in vitro cell culture is the bulk population culture. To further validate their in vitro culture result, they culture the single cell colony that isolated from each population under the same T cell induction medium and try to validate that this 6 population of the cell can recapitulate the trajectory. After they culture the cell for 7 days, they find still the single cell isolated from the population 6 go into DN2 cell fate easily. But the population 1 have the lowest speed to enter the end stage of the T cell lineage. So that means the cell have successfully recapitulated the pseudotime analysis. That is, the cell that express the marker of the mature ETP cell can enter the DN2B stage very easily. And they're also trying to isolate the single cells from the 6 population and to reduce the 10x chromatin single cell sequencing. And after that, they try to map the transcriptome expression pattern along the pseudotime analysis to see which one is more advanced toward the end stage. And after they map the 6 population into the trajectory analysis, they find still the ETP population 6 have the most advanced status along the whole trajectory. When they map to the pseudotime, they find only the ETP population 6 and 4 have the more advanced status toward the end time that compared to the DN3 populations. If we map the transcription pattern of these 2 populations going to the detailed genes, we can find only these 2 populations express the intermediate genes that identified by the previous single cell sequencing data suggested as intermediate wave genes control the transition step of the T cell lineage. So this analysis just showed that even using the new markers that identify from the single cell sequencing, the population can still recapitulate the pseudotime that reviewed by the single cell sequencing before. So here comes to the conclusion and open question. Following the logic flow of the central questions, we successfully digested the complicated study into several steps and answered each question. So firstly, they answered the question that the ETP population isolated from the thymus is not that pure. There are some non-T cell lineage contamination in this so-called ETP population. And then they used bulk RNA sequencing to identify the differentially expressed gene between the ETP and the DN2. And then they also used the in vitro cell culture to find there are a few of the cells may go into the granulocyte rather than the ETP cells. And after that, they used two single-cell sequencing technology to identify the outgroup of the ETP cells and also find the co-expression pattern of the stem and the T cell genes. So that means there may be some transition stage between these two distinct stages. They are trying to use the classical single-cell sequencing technology to find the correct order of the T cell development. They use the analysis to find out the cell changing directions and also use the DDR tree analysis to find out the trajectory that the cell may take. So after that, they use the velocity analysis to find out differentiation status the cell may take, and use the in vitro cell culture and the single cell sequencing to confirm that the zero time they find out is true for the real transition of the T cell lineage differentiation. Here comes to their conclusion. Using 3 different platforms of the single-cell sequencing, they firstly find that the sequential substage within the ETP compartment before transition to DN2A, marked by asynchronous downregulation of progenitor genes and the transient activation of the gene waves. Firstly, we know that there's no branching during the transition. Instead, it's more like a step-by-step transition that some genes may appear here, but appearance of the non-T cell genes, that doesn't mean they have several branches, but means they are like an intermediate gene wave that control or prime the cell for the next stage. Except for the monotonic increase and decrease of the stem cell and the T-specific genes, there are several multi-lineage priming and gene expression waves that control the intermediate stage of the T cell transition. And even for the stem cell and the T-specific genes, they are not appear or disappear at the same time. Instead, they will disappear at different times. Some revoke very early and some disappear after T-specific lineage genes appear. So it's like a partially overlap the pattern of the stem cell and the T-specific lineage genes. The second conclusion is that the frequency of T lineage potential is very high in ETP overall. And although some transient activated genes are otherwise associated with non-T cell fate, alternative lineage potential in pro-T cells decreased monotonically as the cell progressed from the FLT positive ETP to FLT negative ETP to DN2A to commitment. So this is also consistent with the developmental hypothesis that along the differentiation and the development of the cells, the multipotency of the cells will decrease, and they have lost their ability to give rise to several lineage. But instead, they have higher capacity to go into the mature stage and contribute to the end stage powerfully. Here comes to 2 open questions. The first is that whether we can apply such analysis to our own study. So you can try to think about your own field and to see whether there's any questions that has not been answered yet. This study is more like a pipeline, and you can try to follow it to answer the questions in your own field. The second question is that whether we can use the SeqFish to map the RNA expression in tissues. And the answer is yes. So now this group have developed a very new technology called the SeqFish Plus. And this is my presentation. Thank you.